Hello, for those who don't know me, I'm Dr. Brent Vaughn. I love working at 4Life because I find transfer factor science so fascinating. There is so much to learn and so much we already know. Today, I'm going to talk to you about some of the science so you can become a 4Life transfer factor expert. In order to understand 4Life transfer factor, it's important that we first understand the immune system. As you can see, the immune system is actually a bunch of different organs spread throughout the entire body. The biggest parts of the immune system are those found within the gut, the bone marrow, and your lymphatic system. In a very real sense, the immune system covers the entire body. You have immune system cells in or around every tissue, organ, and structure within your body. Having a healthy immune system helps maintain the health of every other organ and system, from the nerve cell at the top of your brain to the tiny blood vessels in your pinky toe. All of your immune system cells are created in the bone marrow. It's a spongy substance found in the center of your long bones. Shortly after immune system cells are born, a portion will migrate to the thymus where they mature. The rest remain within the bone marrow until they are ready to enter the bloodstream and find their destination within the body. About 70% of your immune system cells end up in your gut. Another large portion are located just under your skin or along the mucous membranes of your body, places like your lungs, your eyes, your mouth, nose, and throat. These areas of your body have many immune system cells because they are the sites where an infection is most likely to start. Therefore, the body concentrates immune system cells in these locations to protect and maintain your health as much as possible. The lymphatic vessels are like a second circulatory system. In many places, these vessels run parallel to the circulatory system. Lymphatic vessels serve two purposes. First, they are a conduit for some bodily waste and some of the fats from your food. Second, the lymphatic vessels are a dedicated highway for immune system cells. These vessels are another way immune system cells can move throughout the body from one organ to another or from one lymph node to another. On that note, lymph nodes are concentrations of immune system cells located in key areas around the body. The most well-recognized of these areas are your neck or under your arms. These locations often swell when you're sick. This swelling is a consequence of the rapid expansion of immune system cells as your body mounts a response to the infection. I mentioned the large number of immune system cells located within your gut. Most of these are found in areas within the large and small intestines called Peyer's patches. These concentrations of immune system cells are the hubs within the gut where your immune system monitors what you've eaten or things that have otherwise entered your mouth. These immune system cells work in a close relationship with the friendly bacteria called probiotics that also live within your gut. These two groups work together to hopefully prevent any dangerous organisms from getting into your body through the gut. When your gut is healthy, this symbiotic relationship works well. The probiotics provide a ground cover of sorts that makes it difficult for anything else to find a foothold and begin growing within your gut. Your immune system cells are constantly patrolling for things that don't belong. When these immune system cells encounter something that shouldn't be there, they work together to remove it. A healthy immune system and a healthy gut work hand in hand to help maintain your overall health. We've moved from the whole body to the gut, and now we're down to individual immune system cells. These are the main types of immune system cells. There are several subtypes within each of these classes. There are hundreds of different immune system cells. Each type and subtype have a specific, or rather several specific, functions and specialties. In a lot of ways, you can compare your immune system cells to soldiers in an army. You have many different soldiers. Some are not very specialized, while others are highly specialized. T cells are like your general infantry. They're the ones that do most of the fighting. However, there are some specialized T cells that function as commanders and other specialists within the infantry. B cells are intelligence and reconnaissance officers. They identify and mark the enemy for the immune system cells. They produce antibodies that perform this function, and they limit the infectivity and activity of invading organisms. Natural killer cells, or NK cells, and neutrophils are your snipers and special forces. They are often the first soldiers sent in and have specific targets. NK cells specifically target defective or virus-infected cells. NK cells and neutrophils are the first immune system cells on the scene. You can think of macrophages as tanks. They swoop in and ingest invading organisms. They are larger cells and are less focused on any one type of invader. Each of the other cell types has a unique role in the overall immune system response. When each does their job and they all work in well-coordinated fashion, you stay healthy. Sometimes signals may get crossed. Some troops don't show up for the battle, or the wrong troop arrives at the wrong time or wrong location. When that happens, you may get sick. Thus, it's important that all the cells work together, each doing their job in a precisely executed operation. The immune system is a vast, interconnected network of cells and organs, all working together to maintain your health. Keeping this system running at peak efficiency is vital to maintaining your overall health. Now let's dive in a bit into a brief discussion on the history of transfer factors. There's a little more than 70 years worth of research behind these small molecules. Even with all that time and the millions of dollars spent on research, there's still a lot we don't know about these tiny compounds. The transfer factor story starts back in the mid-1940s with a gentleman by the name of H. Sherwood Lawrence, a medical doctor and researcher at New York University. At the time, infectious diseases were a major public health concern, kind of like today, actually. Dr. Lawrence noticed that most people infected with an illness didn't get it again, at least for a time. He theorized that there was something these people had that protected them. He wondered if it could be transferred between people. Was there something in these already infected people that could be used to prevent illness in someone else? He decided to start looking, probably in one of the most obvious places, white blood cells. White blood cells are the immune system cells circulating within the bloodstream. There are dozens of different types of immune cells circulating within your blood at any given time. Dr. Lawrence took the white blood cells from a person who had survived an illness and injected the white blood cells into a person who had never been exposed to the illness, what we call a naive person. He waited a couple days and then did a simple test to see if the previously naive person now showed signs of an immune system response. Sure enough, there was an immune system response. He then repeated this experiment, but this time he broke open the white blood cells and gave the contents to the naive person. Same cells. The naive person now showed signs of immunity. Fast forward a few years, and Dr. Lawrence kept refining the process until he determined a specific fraction of the white blood cells content that was responsible for the transfer of immunity. He then coined the term transfer factors, as there were factors within the white blood cells that could transfer immunity. This research caught the attention of many other researchers, and the heyday of transfer factor research began. After Dr. Lawrence's first publication and a few additional decades of research, we knew a bit more about what transfer factors did and how they could be used. Unfortunately, the technology of the time was the biggest hindrance. There simply weren't a lot of options in studying really small molecules in a living organism. They made a few good efforts, but really only ended up creating a slightly tighter definition on the molecular weight or size of the transfer factor molecules. Efforts to identify the building blocks of transfer factors were really without success. Let's step away from the history books and take a minute to put the size of these molecules into perspective. You can see a numerical and somewhat pictorial representation of just how small these molecules are. We start our journey with an average woman weighing about 60 kilograms. Next, we move down to a melon with a weight of about 1 kilogram, so about 60 times smaller. Next, a 1 gram paperclip. That's a further 1,000-fold reduction, or 60,000 times smaller than our woman from the beginning. Now we move to a snowflake, weighing about 1 milligram. Now, for those of you who aren't plagued by snow each year like we are here in Utah, think instead of a single grain of rice. However, we need to cut that grain into 30 little pieces. Now we have something that weighs about 1 milligram, 60 million times smaller than our woman from the beginning. That's not nearly small enough. Let's throw 10 more zeros onto that number. Now we're down to the size of a single transfer factor molecule. For those of you keeping score at home, we're down to something around 600 quadrillion times smaller than where we started. That's a very big number for something so very small. So what does that mean? It means that inside every transfer factor capsule, there's room for trillions of transfer factor molecules, enough to educate a good number of your immune system cells. So what are transfer factors? In a nutshell, 4 life transfer factors are small proteins, peptides, and other compounds concentrated from cow colostrum and chicken egg yolks. Back in 1989, 2 transfer factor researchers found transfer factors in cow colostrum, and they patented a way to concentrate them. In 1998, David Lisenby found and licensed that patent. 4 Life was born. A few years later, under the direction of David Lisenby, 4 Life identified transfer factors in chicken egg yolks. We patented this process and later patented the combination of these 2 transfer factor sources. These 2 sources offer some potential for additive, if not synergistic, benefits. Colostrum is the milk produced by mammals in the first 1 to 3 days after giving birth. The yolk of an egg is the sole nutrition source for a growing chick embryo, and its only chance to get immune system protection from its mother. Whereas the cow can continually pass on small amounts of transfer factors in its milk for as long as the calf continues to nurse, the chicken has to pass it all its information when the egg is laid. Therefore, the cow provides a wide variety of transfer factors in its colostrum, trying to cover as many possibilities as it can. A little bit of a lot. The chicken is much the opposite. A lot of a few things in an attempt to heavily protect the chick from the greatest threats. Can you see why combining these 2 has such great benefits? The process of collecting 4 life transfer factor molecules is rather simple. 4 life transfer factors are obtained from cow colostrum through a largely mechanical process. Don't worry, newborn calves are given all the colostrum they need before we collect the excess. It is just simple filtration. No chemicals, nothing gets changed. We just concentrate these small compounds by running the whole colostrum through a series of separations and filtration. It all starts with removing the fats from the colostrum by spinning the whole colostrum at very high speeds. The resulting liquid is then passed through a very fine filter that only allows compounds less than 10,000 Daltons in size to pass through. Remember our size graphic from a few minutes ago? 10,000 Daltons is the currently accepted size for transfer factor molecules. We use a second, even smaller filter for nanofractor molecules. In this case, the filter only allows compounds smaller than 5,000 Daltons to pass through. The colostrum liquid is passed through these filters a few times to concentrate as many molecules as possible. The final liquid is then sent on to a reverse osmosis and spray drying process to remove the water and turn the liquid colostrum into the powder we're all familiar with. That's it. There's still a fair bit of science behind the filters, liquid flow and fat separation, but you have the basics. The whole process is all done without modifying the transfer factor molecules and without the use of any chemicals. Oval factor goes through a similar process. In this case, we use a phase separation technique to separate the yolk-based transfer factors from the whole egg. This process is identical to the colostrum concentration in that no chemicals are used and the transfer factors in the egg yolk remain equally unchanged. That wraps up our discussion about what makes 4-life transfer factors unique and how we can get them from cow colostrum and chicken egg yolks. Now for all the exciting futuristic stuff. This is the part of the discussion today that gets the whole research and development group out of their seats. Feel free to jump up and celebrate with us. Just don't spill your drink in the process. There have been many studies conducted on transfer factor over the years, and many of you are probably quite familiar with some of our earlier studies. Today, we're going to focus on some of our newer research that may be unfamiliar to many of you. If you want to travel back in time a bit and revisit some of our earlier studies, I encourage you to do so with the TF certification course on the 4-Life Academy. The science is still solid and still fascinating. Now, don't adjust your TV or computer monitor. It's not fuzzy, and you're supposed to be seeing dots. This is a study using new technology called flow cytometry. Now, that's a rather fancy term that probably means nothing to most people. Let me explain. This machine uses very small tubes to allow cells to pass by the laser detector one cell at a time. This machine can count every single cell and measure several aspects of that cell's activity. What you're seeing here is the output from this machine. Each dot represents a single cell. The red dots are the NK cells. The green dots are the target cells. The panel on the left is the baseline. This is the number of NK and target cells before the batch was incubated with transfer factors. The panel on the right is what they looked like 48 hours later, after being incubated with transfer factors. Not only are there fewer green dots, there are a lot more red dots. This tells us that the NK cells were able to kill a good number of the target cells, and they also dramatically increased in number in just 2 short days. This is a very precise way of measuring NK cell activity. Furthermore, using this method, we can investigate not just the type of cell, but some indicators of its activation and the compounds the NK and other immune system cells produce during their response. We spent the last several years working on this method. There are still a few more refinements we need to make. Once we have it all nailed down, we can use this method to look at other immune system cells and see how transfer factor affects those cells over time. What happens in the first few hours? What happens after a day? Which cells respond first? Which respond next? And so forth. Hopefully you see the exciting potential this technology has. We're certainly excited for the future of this and related methods. Let's move on to another recent study. In this study, we looked at the compounds produced by activated immune system cells and the activity of another immune system cell type called macrophages. Here, we just looked at transfer factor plus. Adding transfer factor plus led to a 22% increase in macrophage activity. Remember how I likened macrophages to tanks? These immune system cells literally eat invaders. They are large, non-specific vacuums of the immune system. Their role is to physically remove invaders and debris from the site of infection. They enter the immune system response somewhere in the middle and are far from surgical in their actions. Nonetheless, they are very important. Again, adding transfer factor plus led to a 22% increase in macrophage activity. On to another item that we looked at in this study, cytokines. Cytokines are a large group of communication molecules for immune system cells. Cytokines are the signals immune system cells use to attract other immune system cells, to stimulate cells, and even to stop an immune system response. Most cytokines have short lifespans and only affect other immune cells in the immediate surrounding area. They usually do not circulate within the blood and are rarely seen in large quantities. You can think of the increase seen here as the immune soldiers all pulling out their bullhorns and yelling to the other immune system cells. Hopefully, everyone hears the message this time. We saw a 36% increase in both cytokine release and the same increase in immunoglobulin G production. So what are immunoglobulins? Immunoglobulins are chemicals produced by B cells. See, we added another immune cell to the TF mix. Immunoglobulins serve two purposes, which I mentioned earlier. First, they mark things for other immune system cells to remove or kill. Second, they interrupt the invaders. Immunoglobulins bind to the surface of the invaders or otherwise get in the way, making it harder for invaders to start an infection. It's easy to see why an increase in IgG can be a good thing. Immunoglobulins are usually some of the latecomers to the immune system response. It can take up to 2 weeks after an initial infection before large quantities of immunoglobulins are produced. Most of the time, it takes several days before levels are even measurable. We've discussed several examples of transfer factors stimulating the immune system cells. Transfer factors increase immune system cells' activity or the compounds they produce. We've also discussed how transfer factors keep immune system cells focused on real threats, helping them avoid responding to something that isn't a threat. Let's put all this together. What we're really striving for is balance. We want an immune system that reacts quickly and powerfully when needed, but your immune system also needs to know when to relax. A perpetually heightened or stimulated immune system will eventually crash. A sluggish or dormant immune system will likewise fail to respond when needed. Neither scenario results in optimal health. I hope that over the last 20 minutes, I've shown you that 4-Life Transfer Factor is capable of both helping your immune system respond when needed and relax when there is no threat. In summary, your immune system has to repeatedly do 3 things. First, it must accurately and quickly recognize things that don't belong in your body that can make you sick. This process can take hours, or it can take a few days to fully complete. Some of the delay is by design. Your immune system has several checks in place to hopefully prevent misidentification of threats. The longer it takes your immune system to make a correct ID, the more time the invading organism has to grow and multiply, thus making the upcoming battle more difficult for your immune system cells. Accurate and rapid recognition is essential for the healthy immune system and a healthy body. Second, your immune system has to respond appropriately to any threats. A weak response will allow illness to linger. An excessive response can be lethal in the worst cases or just debilitating otherwise. Knowing which troops to call in and when, along with effective coordination of the overall response, is critical to our long-term health and quality of life. Lastly, let's not forget the importance of remembering. Perhaps one of the greatest skills used by your immune system is the ability to remember past invaders. While some of your body's enemies are constantly changing, many of them stay very much the same. If it were not for your immune system's ability to remember these past health threats, we would get the same illness over and over again. You usually only get infectious like chickenpox once because your immune system is able to remember that first infection. You can be exposed numerous times after that and not get sick again. Your immune system remembers the virus and it encounters that virus again. The recognition and response start immediately, and the virus is removed before you feel sick. We are all striving for a well-balanced immune system that can accurately recognize, respond to, and remember potential threats. Transfer factors support the immune system in its effort to fulfill these 3 functions. Thank you for joining me for this brief overview of your immune system, where transfer factors come from, how 4Life gets our transfer factors, and some of the cutting-edge research we've done for these amazing little miracle molecules. This just scratches the surface. And thank you for joining us for International Convention Shine 2021.
There's a fire inside and I just can't hide it. There's a fire inside and I just can't fight it. Ooh.
And now we invite you, yes you at home, to spend more time with us by viewing all of our bonus content. Find training information on sales and marketing, product science, safety and regulatory, and more. Check it out.
You know I'm